Hello everyone. This is the last mini lecture in our gene expression series. What's going to happen here is that we have ended the transcription phase and we have our messenger RNA, but we need to modify the messenger RNA so that it can be used out in the cytoplasm. So this transcription was just previous to this, and we have generated the messenger RNA. Now we are taking this messenger RNA, we're going to modify it, and then it's going to be moved out of the nucleus through, these nuclear pores and into the cytoplasm. So the next phase will be translation. Hope you enjoy. Let me get things ready for you here. We don't need those pop-ups. Let's begin.
Messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliciosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids.
Okay, right here is an important point. We have our list or table of codons with the specific amino acid that these codons code for. It's important to know that we can make about 10 of these in our body, and the other 10 we need to take in by eating protein. And then our body digests the protein, and then we can absorb the amino acids. So that's just an aside to this translation process.
There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites.
Okay, let me review these sites with you, and then the narrator can go over it again. The A site is going to be the attachment site. This is where the transfer RNA comes in and attaches to the codons. So the anticodons will attach to the codons from complementary base pairing. Okay? Then once that occurs, the whole messenger RNA shifts down into the P site, and this This site is for the peptide bonding to occur, P, peptide, okay? The little amino acid here will bond to the amino acid that's going to be coming off of the transfer RNA from the A site. So the peptide bond will build the polypeptide, the protein. Okay? Once that occurs, this transfer RNA no longer has an amino acid attached. The whole thing shifts over here to the E site, and then the transfer RNA that does not have the amino acid, it is ejected, or it exits, and it needs to be charged up with another amino acid. So that's what happens here. Attachment, peptide, bond, and eject or exit. Okay?
During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation.
This is a polypeptide, another name for a protein.
The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract.
Okay, now if a protein that has been made through translation is secreted out of a cell in a vesicle, then that is called exocytosis. That was part of the active transport mechanism way back when we learned about the cell membrane and the transport mechanisms of the membrane. So just to review, that's exocytosis, because it's a vesicle releasing substances, secretion through using a vesicle, secreting through the membrane. That's exocytosis.
Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
Now, this was a very informative video, and we see here that if we take food in, proteins break down the food, and these proteins are known as enzymes. They digest the food into chemicals that the organs can absorb, and then these nutrients become chemicals that we can use for building DNA or building proteins or building fats, you know, all sorts of things. So we need all sorts of chemicals to make different molecules within our bodies. Hope you've learned something here, and have a nice day.